Hello. Are you getting me? Hello? Are you getting me? Guys, unmute, please. Okay. How are you doing? Guys, can you unmute? I don't. We want the class to be. How many people do we have connected? Right now we have 8. We are still waiting for order. There is someone who is new. There is someone who is new. I mean, the person. If there is someone who was absent last week, because last week was the first session. Okay, people are connecting. Hi, guys. We still have like 5 minutes. No one has a question to ask. Guys, please, can you do me a favor? I would like you to unmute so that we can interact, please. Because when there's not interaction, the class doesn't sound good. I want us to interact. Can you put on your I said I want you guys to unmute. I want you to interact vocally. Unmute right now. Excuse me, not right now. Let's say for the moment, because we are still waiting for other students. If you unmute, at least we can interact, and you can get to know also each other. Because when we are like that, it's like, there's no interaction. I want the class to have a good life. Okay, it seems like I am having new face like Hamba.
Good afternoon.
Good afternoon.
Hi.
Hamba, good. Hamba, where are you located? In which country are you attending this training? From which country? Are you getting me? I think
Good afternoon.
Good
Sorry,
afternoon,
this
guys.
is Professor.
I am seeing Perpetual. Perpetual, from which country are you attending this class?
I'm from Zambia.
Okay, were you present last week or not?
No, I wasn't.
Okay. Anyway, last week was more just presentation. Today is the day thing will start really.
Oh, okay.
We will start explaining key concept and
Good afternoon.
good afternoon, my friend. It's Pidio. I hope I am pronouncing it well.
Okay, yes, it's Pedro actually, but it was wrongly spelled by Google then, so I decided to leave it. It's Pedro. I'm calling, attending this workshop from Nigeria. It's nice meeting you.
Okay. I'm seeing Ali is asking, is the preview lecture recorded? Unfortunately, I did not record it. I think I will try to record this one. Gérer l'enregistrement. Yeah, I will definitely record this one.
Good afternoon.
Good afternoon, guys.
This is Antoine
It is
from
Antoine.
from Democratic Republic of Congo
Mm.
in eastern of DRC in
I know
Bukavu.
Congo. I know Congo.
Yeah.
I have
Okay.
never been there, but at
Uh,
least when I was in Belgium in the past, I had a lot of friends from Congo.
oh, thank you.
Okay.
Thank you. Thank you.
It is 10:30. Let's wait maybe for 10 minutes to have order, or 5. I think a lot of people are still connecting. Who was absent? Yes, last week. I think there are some new face. There's some new face. Okay, guys. First, I am not an expert in PowerPoint, but I'm just expert in transmitting knowledge. So if you don't see very nice PowerPoints, just be focused on the knowledge. In the knowledge you will receive, that's the most important thing. In 5 minutes we will start. Let's wait and see if other will join. Who said Maki? Someone is saying this is my first session. Who is it? Let me see. Malak. Malak Hani. Malak, where are you attending from?
Hello, my name is Malak Hani. I'm from Egypt.
From Egypt. Okay. Say hi to the pharaoh from mine. I'm seeing Lassina Traore. Lassina Traore. Where are you from, Lassina Traore? Someone is asking if the certificate will be delivered. As I said, unless I start charging, I can deliver certificate. Maybe from the next session. Let's say the next session. Yes, this is the first session. I did not plan to deliver the certificate. And as I said last week, the most important here is not certificate, because you can have certificate without knowing anything. Certificate is just to show that I have attended this training, but maybe I have very American perception of certificate. Here in America, for instance, we don't care about certificate. We care about what you're able to do. So what I can suggest to you guys is to create a GitHub. The GitHub is something that can allow you to show to people on LinkedIn that you have done some project in next generation sequencing. It's better than a certificate. Certificate means nothing. It's just a paper. But when you can copy the link of your GitHub with the project you have done in it, when you share with maybe an employee or with a supervisor, it gives more trust because we will be seeing what you have done before. So I know that in the African culture and also in the Indian culture, I'm African. So when I say African culture, guys, I'm African from Cameroon. But in the African culture and in the Indian culture, people like a lot diploma certification. But I am here to tell you guys, the job market don't care about certification. The job market care about skills. And the best way to show your skills is to have a GitHub. That's why I want this project will be very hands-on. It's not theory, it's very hands-on. And I would like you guys to have a GitHub. And if you don't have the GitHub, we can organize a class, a short session for that to help you so that at the end of this project, you will be able to say somewhere, I am able to do an RNA-Seq analysis and show what you have done. Okay, we are 10:35 Boston time. Guys, let's start, okay? Let me share my screen with you now. And we start the job.
Okay, thank you very much.
Are you with me?
Yes.
Are you seeing the screen?
Yes,
we
we
are
can.
seeing it.
Okay. Again, I want this class to be interactive. If you have a question to ask, don't hesitate. If I don't know, I will be honest to tell you that I don't know, and together we will go through it. If I know, I will teach, I will tell you. Because in the American culture, teacher is not a god. It can happen that he doesn't know something, and he should be honest to tell you that he doesn't know. And together we will go through it. So that's American culture. Okay, guys, again, bulk RNA-Seq. When I say beginner, normally this training is more than beginner because the skill that I will be giving you, you can apply it in any circumstance. Okay. For those who was not here there last week, what is RNA-Seq? What is RNA-Seq?
I think with my little understanding, RNA-seq means sequencing RNA in order to get the nucleotide arrangement of the RNA.
Perfect.
For
You gave the simplest and the best. Even my grandma can understand exactly what you are saying, and that's the best way to explain science to people, using short words, simple words to make yourself understand. Okay. As we said, yes, last time, is to give the sequence, to determine the sequence, the alignment, the arrangement, the order of nucleotide in the DNA or in the RNA. That's what we call DNA or RNA sequencing. And today we will be focusing only in the RNA-Seq. Here we will be working with a reference RNA-Seq. Oh, I did a mistake, guys. Sorry. Okay, it's a bit like this, right? As you can see, who can tell me different step to process the sample from the sample you have, biological material, till data analysis? Who can tell me different step? Can someone give it a try?
yes,
Okay, go, my friend.
you extract the RNA.
Yes, I am asking what are different steps. What are different steps in the RNA-Seq process? So from the biological material till the data analysis, so end to end. For the end-to-end project, how can you list different steps?
Yes.
Okay,
yes, I
go
think
on.
the first step is first to isolate the RNA.
Yes.
Then, if you are interested in a particular messenger RNA, you try and amplify it to make it large quantity by doing PCR.
Okay.
Then thereafter, then if you are able to get the size of the messenger RNA you are interested in, then you can now send for sequencing. Then during the sequencing, you prepare your library, and after the library, then you sequence depending on maybe you are using a first generation or second generation sequencing.
Okay. Yes, and after that, you have your data ready for analysis, right? So we have two main steps. We have two main steps: the lab step and the computational step. The lab step go from the isolation of your genetic material from the cell line. As here we have the cell plate. We isolate the RNA, and when you isolate the RNA, they say here the fragmentation, but sometimes you can make first, you can convert your RNA. Let's say if you want to analyze the expression of some gene, because we have different type of RNA. If you want to analyze the expression of some gene, you can convert first your RNA into cDNA. We call that reverse transcription. Then you send to the, if you have a sequencing platform, you create the library and you sequence it. This is the wet lab part. When you finish sequencing, you have what we call the read. This is where we will be acting. People have already sequenced. They have already designed the experiment. They sequence the data. So your work as bioinformatician or as genomic data scientist is to analyze and to find the information that you need. Maybe it is the differential gene expression, mainly in the reference RNA sequencing. Guys, what is the meaning of the word reference here? It's very important to know that. Very, very important. And in the job interview, they can test you in that. What is the meaning of reference in this RNA-Seq data analysis? This word is very important. Can someone give me a try?
I don't want to try.
Yes.
I think it's like a consensus sequence, as in the one that everybody has, the bioinformaticians have agreed to be the one that can be used to compare any other sample. That's
Thank
the
you.
little understanding.
Thank you. And you can see it here. When you sequence, you have the read. The read are short fragment of your sample, of your DNA or the RNA of the, let's say, of your gene of interest. So you have here the reference. You will align the read to the reference genome in order to see if they match. You see here, it should match. Different portions should match. Here is the reference genome. And then you quantify to have the expression profile. This is the principle. It's the same explanation. You have read and you have your reference genome. You map them. Here is your reference genome, and you have different read that will be used to map, that will be mapped on your reference genome. And you can see here you have different fragments. You compile all these different fragments to have this. Okay, some people can call that quantic but let's say read 1. It's the larger portion. You had the smaller portion here. You align them together to make the first big portion. The middle part, you have some read, 3 read. We pull them together. We align here, and we have the third part with 1, 2, 3 different reads. You know, at the end of the day, by combining all those reads, we will be able to align with the reference genome here. That's the principle behind. What is the opposite of reference RNA-Seq, guys? If we have reference, it means that if we have reference RNA-Seq, it means that we have another one who is not, that doesn't use a reference genome. True or false? Guys, say something. True or false? Are you with me or not?
Yes, we are with you.
I am asking one question. If we have reference RNA-Seq, it means that we have another version of RNA-Seq that is not, that doesn't use reference genome, right?
Yes.
And how do you call that technique?
That's
Is it de novo?
Thank you. It's a de novo. In the de novo,
De novo,
you do the same thing, but you don't need really the reference genome for your mapping. Thank you. Now let's go in the first part of the job. I will first ask, which operating system are you using, guys? People who has Linux is fine. They don't need to do something. But if someone is using Windows, we will be installing WSL. Without that, it will be difficult for us to manipulate, to handle file. Because WSL, it's a Windows system for Linux. So it allows us to run Linux command in the Windows operating system without any need of installing a new operating system in our computer. Are you okay, guys?
Yes.
Can we move now in installation first?
Yes, we can.
Okay, let's go in the installation. First, I want you to type command. Command line. I think I can give you this. I can give you this. Let me give you this comment. I will give in the chat. To give you this comment in the chat. Are you seeing the screen? Are you still seeing the screen?
Yes.
Where do we have our chat?
After the participants.
There is someone making a lot of noise. Please, we have to respect each other. Can you turn off your if someone is busy doing something, can person turn off his microphone, please? Guys, who can guide me? I don't even know where is the
chat is after the participants. If you check towards your extreme right, then you will see something look like a triangle.
Oh yes, thank you. It is no, this is my WhatsApp.
No, towards the extreme right. Is the
Extreme
second
right
to the last
here?
icon. Yes.
No.
Before the 9 dots.
Here is should I go back?
Sir, you
I
should
just
go
forgot.
to Chrome, sir.
No, 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 no. It's not, you are not using your full screen. Assuming you are using your full screen, then
Okay.
I'll be able to show it on.
Okay, let me see. Here. I want to give you this line of comment. Otherwise, it will not be interesting.
Okay. Do you see those the lines where you have the microphone, the video, and
No,
some of that?
no, I don't see it. I don't see them.
Oh.
I don't see them.
We are
That's
both,
why. Can
but
someone
we are
try to
using
type?
this.
Can someone type a message or type something? Maybe it will appear and I will see.
Okay.
No,
Okay.
no. I I
You are
think
not
that.
seeing?
Okay, I am seeing something now. I'm seeing. Thank you. Let me go back. Okay,
Yes,
I'm
good,
sharing
good.
comment with you
Okay.
guys. Can someone? My suggestion is that you open a notepad, you paste it there, so you can just make a copy paste. We will do it together. Okay. Okay, are you ready, guys? You type first comment prompt. We can also do it with PowerShell, but I am more comfortable with comment prompt than PowerShell. I know that a lot of you have never worked with command line. I know, but we will go slowly so that everyone will be able to execute his first command line without clicking. Are you ready? Are you here?
Yes.
If all of you are here, please let. I want to be sure that everyone is comfortable. Are you all there?
Yes.
Yes, people are responding. Yes.
Okay, perfect. Let's move to the next step. You type WSL. What's going on with my system? Okay. When you type SL, what do you see? What do you see, guys? For me, I have already WSL installed. That's why we will not have the same thing, but I would like you to, if one of you can share the screen, I think it will be great. So that the person, you will be seeing what the person is doing. Hello? Can someone take the lead? I will be just guiding the person so that, because I have already installed, I want people to see how it works in the real condition. Okay, I will choose one person since you don't want to. François Xavier Dembele. Is François Xavier Dembele with us? Guys, who would like to take the lead so that I will guide you, because I have already that installed and I cannot guide you if I am not seeing the screen of someone. Am I allowed to speak those guys with me? No one want to try? Oh. again. Okay, Timo. Okay, let you will share your screen. Let me stop sharing mine. Take the lead. Who can share the screen with us? Okay, perfect. My job will be just to, okay, when you type WSL, what do you see?
So these are what I saw: argument, install, option, then argument example, import, no description, then app, list, option. So these are the things that's on the screen.
Okay. Anyway, it is showing that you don't have the application installed, right?
All right, sir. Sorry, you are muted, sir. We can't hear you.
You type what you will copy the second comment. You paste. Just copy and paste. Don't type. Something will happen. WS space hyphen update. Just copy and you paste. Everyone should be doing the same. I'm just using his screen to guide you guys. Okay. When you type WS update, okay, you are doing well. Just continue. Copy the other. If someone is facing any issue, please, guys, let me know, because we will not come back. This is the critical part. If you don't have this installed, you will not be able to work. Is it going well for everyone?
Okay.
Okay, someone is facing problem. Someone is facing problem. We have to stop and solve the problem before moving on. Can you put a full? Guys, do you see the screen of our teammate Tomilola? Are you seeing his screen or not?
Yes.
Are you seeing his? Okay.
Yes.
It means that everyone is doing great, right? You just execute. It is so everything was done to make for me it indicate was not installed. Yes, for the first comment, you will see it will say that it's not installed, but when you will run WSL update, the process will start from there. I send you all the comment. It's the easiest way to install. So If someone is facing hmm, they're saying that a lot of people want to join, but I don't know how to add them because I have been adding people. Can someone help me to add other? Is it going well for everyone? Guys, if you are doing well, if it is going well, give a thumbs up. I want to make sure that everyone is satisfied. Personne souhaite. Who has finished? I shared the prompt in the chat. Some people are asking for the comment. Someone who is typing, I don't have, if you have a problem, please voice it because you are a lot, we are about 40. I cannot go through 40 comment. If there is something to say, voice it. Please. Does the map?
Excuse me, sir.
Yeah.
If somebody have Linux as well as Windows on his system, do I still need to install this? Since I
No,
have a
no. We
okay.
install. That's why I said if someone, because I know that most of people use Windows. That's why WSL is useful because it allows us to install Windows. It allows us to install Windows to run Linux command on Windows.
Okay, thank you.
There is someone who are leading.
Sorry, I was open from my mobile. Then when I open the laptop, the command disappear. Can you write it again?
Yes. Let
Thanks.
me copy it. Let me copy them and send to you. I send in the chat right now. Mr. Timola, did you Timilola? Did you finish?
Can you see my screen? I think it's still booting, installing the Windows subsystem for Linux. So it's still at 0.0%.
Where are you? In which part of the comment are you now?
Okay. Hold on. I'm trying to install the Ubuntu WSA install the Ubuntu 22.04.
I understand, but I want to know exactly where are you? Okay, you are now at the level of Ubuntu.
Exactly.
Okay. Okay, let me see. I think that part is very critical. I will explain to you how to do it. Because the version of Ubuntu matter a lot.
All right.
Guys, where are you? Okay. It seems that a lot of people has connected, but they are not in class with us. So I don't want to bet in the number of people who connect. If I have just 5 people who make the class good, is fine. I don't want people to come connect and disappear. I don't want that, guys. If you come here, it's to learn. I don't want people to connect and go because I pay to host this training. I lose my time. And I want that even if I have 5 people, if they learn well, I will be comfortable instead of having 40 people and some are not online. So if someone is not online, I want the person to disconnect and leave the class. Please, guys. I want to make sure that everyone is online. Can you put on your camera, please? Because if you are not online, I will be losing my time for nothing. Okay. Did you finish installing?
From my end, I was having a network challenge, so I have just joined. This is Precious from Malawi.
Okay. I don't know. Let me share a video, right? Guys, I think if I share this link with you, it will guide you better. I share just to comment, but it seems like some of you need more. Let me share a video with you, and I will give you like 15 minutes to watch it and do exactly. It's like 8-minute video. Mr. Tomila, I will take the lead again because I think that some people are facing problem. I share my screen.
All right, sir.
I said I'm going to share my screen again. With Mac, you don't need, Mac is already Linux. You don't need to install. With Mac, you don't need any installation, any. Okay, guys, watch this video. This is only for people who use Windows. Windows 10 or Windows 11, it will work very well. I just share a YouTube video with you. This link I just share with you guys. We don't create anything. Everything exists online. If you know how to do or where to find the resources, you don't have to invent something. That's why I share that video with you. Where are you guys? I give you 15 minutes maximum to watch that video. After that, we will move to the next step, downloading the sequence and start the analysis. Is it going well? Guys, you are all my friends, okay? When I say friend, it means when you are together with me in this class, don't think that the teacher, we are all, what should we do? I said we should be installing the WSL. We should be installing the WSL. I have it already installed. That's why it will not be interesting if I take the lead. But yeah.
Of course,
Sorry,
from my
I
end.
got to.
From my end, I think it's just had to do with my device, laptop, because my laptop is failing to even to restart. So I'm using the phone now, but the laptop is not obeying the instructions. So that's the only problem. But I'm trying it. Once it start picking, because I'm trying it. Once it start pick up, I'll be able to do it.
Okay, it means that you have the problem with your system. But the video that I share with you with the comment, guys, it is the best and the simplest way. But you can also download it from internet. But I want you to get used to command line. I want you to get used to command line, because I don't want to teach you the bioinformatics that will not be useful for you. That's why I did not show you the easiest way, that is to go and click and install. But you can find it online. I want you to use the command line, because this class will be command line, command line, command line.
Yeah, from my end, I'm trying hard so that I'm able to.
Did you see the link video or the link YouTube link I share in the chat?
In the chat. And the unfortunate part is
it is
the chat, because I've joined the class through while I'm trying to fix up my issues with my laptop.
Okay.
So I've joined. I was afraid of just doing, so I have joined using.
Okay, your laptop has the problem. I understand. Let's go to Pedro.
Kindly share the command prompt again, because I dropped off and I can't seem to retrieve it from the chat.
Okay, I share it again. That is the third time. You don't have, you just go again. You come here. Let me share first the command prompt. In the chat. Oh. Okay. I share it right now. You open you go in the search bar, you type comment in the search bar. When you will type that, it will open your comment prompt. You just copy, you paste, you copy, you paste. And if you don't understand, you watch the video.
Should I restart the PC after Ubuntu download?
Pardon?
Should I restart my PC after Ubuntu download?
I don't think so. But if it asks that, don't. Okay, that's a good question. That is, guys, let's put this one on hold. We will do it at the end of the training, because it is taking more time. But we will do it. We will do it. Let's continue. Can we go? We will do that at the end of the program. So the most important part. Now we are starting the pipeline. So we are starting the RNA-Seq analysis. The first part is the data collection.
Okay.
The data collection, that is the first thing. Because the installing stuff, let's say you already have your Linux or your machine, your personal computer with WSL. But guys, please put off if you are not using your microphone, shut it down, because it's okay. First part, the collection. Where do we get the raw data? to analyze. Because when we sequence here, the raw data after the raw data is sent somewhere in the database. Here it's where we are. We are starting now here. Have someone used NCBI in the past?
Yes. Yes, I use it.
Okay.
Yes, sir.
Okay.
Yes, sir.
The
I
first.
use it here.
Okay, guys. The first thing, go in your browser. You type NCBI. The first thing that should be done, you type NCBI. This NCBI in the Google. When you are there, you give a thumbs up. We move to the next step because we are now starting what we call RNA sequencing. We have to go and fetch the data, bring them in our bench work. Our bench work is our local machine, and handle those data in from our local machine or local computer. Are you there?
Yes, sir.
Yes, sir.
Yes, yes, sir. Yes.
Okay, can we move? If someone is not there, the person can tell because I want now we will be going very fast. Okay, I just suppose that all of you are there. When you type NCBI, let me do it with you because this course we should be talking by doing. I open my browser. I type NCBI. You click here. We are here. Are you with me?
Yes, sir.
Okay, so there is somewhere
Yes.
all
Yes.
database. There is somewhere here, all database. In all database, we are looking for SRA. SRA. This database, it's where we are going to find our raw data. Is it okay?
Yes, sir.
Yes,
I
sir.
want you to be doing because it is by doing that you understand. It's by doing that you understand. You click on it. The first thing that I would like you to do is to type RNA-Seq data analysis. Just type that. Just type RNA-Seq data analysis. Are you there?
Yes, sir.
Some people are going faster than others. I want to make sure that we are all. Guys, it is here. If someone is not able to understand, it is here.
Yes, sir. It's clear.
You type NCBI, then you come here. There is a mention all database. You click here. You will have a lot of database. You choose SRA. Then you type this one, sequence RNA-Seq analysis. A screen like this will appear. Exactly, it should not be different because it's the same I am having here. I am having the same. I type. I'm having the same. Are you there?
Yes, sir.
Yes, sir.
Okay.
Sir, we type small or capital?
No, it doesn't matter. Small or capital, it will be the same. No, there's not any
Okay.
problem.
When I do RNA-seq analysis, it give me viral metagenomics. But when I do as you do the same capital RNA-seq analysis, it give the RNA-seq of zebrafish larvae. So which one is correct?
Okay, type with let me see what you mean. Normally it should not be a problem because it's not case sensitive RNA-seq analysis. It should not be a problem. Okay, I see what you mean. Okay,
Yeah,
we
it's
can keep
different.
the same. Yeah, I understand. I understand. Type in capital letter. Go in capital letter. Thank you. I did not realize
Okay,
that.
sir.
But the information are the same. Just for the matter of consistency, I want that we have the same thing. Type in capital letter.
I think you're missing data analysis. You're
Yeah.
only writing.
Yes, I type. Okay.
That's why. But it shouldn't matter.
Yes, it should not be, but I wanted to have RNA-seq. Are you there? Can we move on, guys?
Yes, sir.
Okay.
Yes, sir. We can move on. Yes.
Here, when you type, the first thing you have access to this page. You have first organism. Let me show you from here. But it's the same. You can look at your screen. You will see. We have result. You have organism. First, they organize the different sample or different project according to the organism. It means that if you want to work with human data, you should click here to get access to human beings data. If it is from another species, maybe Drosophila or mouse, you will find here. After that, you can see the result. In the result page, we have that search. It found more than 3 million, and just 20 are displayed right now. One thing that you must always keep in mind, you should never try to come in the first page that appear to download the data. Why? Because most information in this first page is for the job that are still going on. If you download the data from the first page, you will have incomplete data because some experiments are still going on for that. That's why you must always come to last here. Are you seeing my mouse? My cursor?
Yes, sir.
You must always, never forget that. Click on last here. So I can go immediately on here. You are seeing it here. That's something, it is very important. Now, let's move to access. Everyone has access, right? When you see access, you have control and public. Can someone tell me what you mean?
Controlled, closed by the author. Need access from them. It's closed data.
Okay. What do you say?
It is a closed data. We need access from the author of the research.
Yes, we need a permission. So it means that it's private. It's not yet public. Yes, it's private. Here public, it means that you can get access from internet. When it is controlled, it means that you need a permission from the owner of, from the owner of those data. Now, so that is, you have source of data. For RNA-seq, it is either DNA or RNA, right? You have data type, you have genome. Okay, we will not go here. You have type, genome. Library, very important concept. Library layout, pair end, single end. Can someone tell me what it does it mean?
So basically,
Yeah, me. Okay.
there. Okay. You can speak if you want.
Very important concept, guys. Very important. And for a job interview, someone can ask you that question. What does it mean, pair N or single N? What is the difference? You must know.
I
You
think okay. Can I go ahead?
okay? Go ahead.
I think pair end means that you sequence both forward and reverse sequence. Why for
Yes.
single end you only sequence maybe either the forward or the reverse.
Perfect. That's it. That's perfect. It means that in the database, when it will be single, you will have in the raw data just one. It will be either forward or reverse. But when you will have per n sample, you will have to download reverse and forward both. That good. Okay, now we have the platform. Platform, it can be Illumina, it's more like the company, Pack Bio. I think for now, it is the most important. These are the most important information that you should know from this in this page. Now, I want to give you the accession number. When you go in what we call, when you go here, when you type SRE, you will realize that any sequence here has an accession number. You can see here. Are you seeing?
Yes.
Any sequence in the database has an ID. It's like your ID. In your country, you have your personal ID that only you have that in the system. It's the same for DNA. Now you will have some metadata like spot, the size, number of base. That is okay. First, I will give you the first thing to do. We will try now to download. I will give you this. So what are we going to do? We will paste this in our what's going on? I downloaded you. One second, guys. Yeah, I type it because this job is what I did myself. Yes, I typed the same. Why it's not working now? Copy, yes. We go in NCBI. Let's start from fresh. I open it. I will share the one it will be working. I will share with you guys. You will be doing the same thing that I'm doing. I just want to make sure that it works. I worked before with it. It was fine, but I don't know why now. It's giving me something. What's going on?
I typed this ERX double one three two
Yes,
double
it's
seven four
fine.
one.
Yes, it's fine. I think, yeah. I think my computer has a problem. I don't know why.
No, I think
Okay.
its site is heavy because I have same result as yours because it's not working. I think site is
Okay.
heavy.
Perfect. Now it's working in my. Let me now share with you. You type this. You do the same. I share in the comment line. Okay. In the this is the accession number. Copy it. You do exactly what I did.
Yes, I did, but now when I go for SRA run selector, click on it and it shows me different results like total run, byte, spaces, and download.
Are you seeing something like this? It should
No,
appear
it's
like
a
this.
description, but my page open like go for its paired and cDNA and complete description.
Yes, you
Where
anyway you must have something like this because it's the same thing I am having here. It must be like this.
yes, actually I click on the send results to run selector, so it show me this one.
Perfect. Does someone having the same? Guys, we are going into a very important concept, and everyone should be able to know how to download after this class, how to get the data from. Is it okay for everyone? Is it okay
Yes, it's
for
okay for me.
everyone?
Yes,
If it is
yes.
for okay, guys, all of you, I want to see yes, yes, to make sure that there's no one left behind. Okay, I just assume that for everyone is perfect. The next step, yeah, maybe someone want to speak. Maybe someone
Yeah.
has a question.
Hello, everyone. So actually, I am getting out with you. So just recap 1 minute what done before I get the meeting. So to understand what happens, because I opened that in NCPI, but I don't know how to download in the command line or download from NCPI.
There is right now. We can download. I want you to download from NCBI because there is a lot of beginner. I don't know if I got your question well, but I
Yeah,
hope
yes.
there is a lot of beginner in this class.
Okay.
We will download the same thing by using command line, but I want everyone first to download the same thing without going with the command line, because for beginners we cannot just start the command line without showing them how they can get the same thing from here. Is it
Okay, so I done. Yeah,
yeah.
done. Download from NCPI.
Yeah, maybe you are very
Thank you.
advanced, I understand, but everyone in the bioinformatics should be able to work to download the data without using command line. When you understand how we can download without the command line, now we will go with command line that is more professional. Not more professional, let's say it's more it is a little bit advanced skill, because you should know the Python. So we will be you should know Linux. We will use command line to run Linux command. That it means that we upgrade. But first, let everyone understand how it works first. Okay, guys, after opening, what did you do? See, okay. Let's move to the next step. I guess that everyone has opened it. When you open one thing, as I said, the organism, you have the organism and all the description. You will see that we have all the information. The platform that they use is Illumina, and it is RNA-Seq for lung cancer. So we will be trying to find the expression, the gene expression between the control sample and the cancer cell line. For people who has worked before in the wet lab, A549 is cell line from lung. So we will be analyzing in this boot camp or in this training the normal cell versus the cancer cell to check the gene expression. Can we move?
Yes, sir.
Yes, sir.
Okay.
Yes,
When you are
yes.
here, the next step is to, I prefer to work directly online so that you will be seeing me. You come here, down, you go down, you scroll down, you will see somewhere run. You click on it. something like this will appear. So it is this. I make the screenshot here. You will scroll down and you come here. You click here. Are you there?
Yes, done.
Can we move to the next slide?
Yes, sir.
Okay. Then you come, you will see that in your screen, you have something like this. There is metadata, analysis, read, data access. You have FastA and FastQC. Download. Click on download. Let me do it as well so that you will not see that he's saying something that he's not doing himself.
Yes, same. Filters done. Page.
Okay. When you click, you will have, you go down, you will see now download, and you have FastA and FastAQ. FastQ and FastA. You will click on FastQ.
What's the difference in it? Fast queue for NGS? Is
Okay,
it?
that's a very good question. That your question is very good. You cannot work as bioinformatician if you cannot differentiate, give the difference between or among different file. FastAQ is more for alignment. FastA format is used more for alignment. And it's, let's say it's a light form. Here, it is bigger and he has what we call threat score. Threat score, it means that the quality score, the quality of the data. Here you don't have the threat score. So you cannot take a FastA format and say how good is your sequencing. But when you have FastQC, you have all the raw data. It means that you have all the parameter to know if the quality, to assess the quality of your sample, of the sequencing. So this one is very large because it has a lot of information that can allow us to know if the quality is good or not. Fastest format, no. Right? There's a lot of difference, but this is the key one. If you give that during an interview, the person will know that you understand very well what you are talking about. Is it okay?
Yes, it's amazing. Thank you so much.
Perfect. And that is a very good question. A very good question. Okay. We are here. No, this is per n. When you click here, it sends you to, I said now you click on FastQ. When you will click on FastQ, it will start downloading here. Are you seeing it?
Yes, same. It's downloading.
Okay. Remember, this is for a single end. I was waiting for someone to ask a question. We talk about single and pair. And I thought that when we will be moving on, someone will come back because there is a big difference when it comes to download pair end and single end. Here is very easy because we are dealing with single end data, right? Now, since he's downloading, let's move. It's when it is working. Now we want to download the pair end. This one is very easy, but you will see that when it will come to a pair end, you will have a problem. Let me share something with you now. For the pair end, you will realize something very strange, guys. My pair end. I extract something. Anyway, can we move on? Can we move on?
Yes.
Okay. Go back to, let me find any sample that, let's go back to NCBI. We have finished with this one. We can just close it because we have finished downloading. We come back to NCBI from scratch. You will see something very important. Guys, repeat the same. Don't say that you have already done it. I extracted a data that I wanted to share with you, but I don't know where I put it.
I have a question.
Yes, go on.
When we go for the quality fast queue reads, then is any reference single or paired? Or if the data is single or if the data is paired, then what will be difference on the result and what the consequencing and outcome?
The reference doesn't care. The reference come, you will never differentiate if it is single or not. Reference doesn't have any problem with pair or single. When you will download your reference in GF, it's GTF format, something like that. It will not tell you if it is single N or if it is single N or pair end. Reference, don't question that. Care just about your raw data, if they are single or pair N. Do you understand?
Yes, I understand. But when we read and data is single or data is paired, if we go for the analysis, then if the no difference, single or paired, then it means we can take both or anyone. And what is its concept?
For sure, for the reference, they sequence both. They sequence both because reference should be perfect, so that if you come with a single end, it can match. If you come with both ends, it can also match at least one brand of your DNA. They sequence automatically both. That's why I say don't care about the reference. Don't care. They sequence forward and reverse in the reference genome, so that if you come with, you can use the same reference to analyze a single end raw data or a pair end raw data by using the same reference. Because when they were sequencing the reference genome, they wanted to have it as complete as possible, because it is the standard, it is the component that will be used to compare. So it should be perfect.
Okay, got it.
Is it fine?
Yeah,
Okay.
thank you.
Guys, let's pick any sample in human being. I want to pick a pair end. Okay. I always say, always go to last, never the first page, because they are not complete. They are still going on. You can come here. We pick any sample. I don't know. If you pick any sample, you click here. You have the accession number here. Some people will either give you the accession number. I don't know where I put the accession number I wanted to share with you. That's why I am coming here. So let me share. Guys, just do like me, but I share first the accession number with you, but do like me. You type SRA, you type RNA data analysis, RNA-Seq data analysis. You click on it. You come, you have a lot, you click on pair N, because now you want just something that is pair N. We choose the species, we want human being. Okay, it will give us a lot of data. And as I tell you, go always on last to avoid taking something that is not complete. Come here, you choose any I click this one. I can share the accession number with you so that you will not have to come to go where to do that like me. Normally, the accession number should be available. Let me share the accession number with you guys.
SR is the not accession number or this data?
Yes, SR is the accession number. I just share it with you. You can copy it instead of typing like RNA-Seq, blah, blah, blah. You just type SRA and then you paste the accession number. It will give you the same thing that I have. Let me come back. If I type this, the same accession number, just see, it will still give me the same. Okay, you can copy the accession number that I sent to you. There is something very important, and always never forget to read layout. Always when you have the accession number, read this parameter layout. When you see pair, it means that you will have forward and reverse. If you don't have that, it means that they did a mistake when loading or uploading the data in the platform. Always you will check this one. Is it clear? Can we move?
Yes, sir.
Excuse me, sir. I am logging in this session. Kindly repeat as given some summary about the term PSS because my meeting is disconnected with that connection.
There is a lot of noise, background noise. I don't understand what you can type maybe. What did you say?
Hello? Can you hear me?
Your connection is not, I don't know if it is the connection or there's a lot of background, so I'm not able to get you really. I'm not really able to get you. You can type. If you type, maybe I will read.
So I am logging in this.
Type it, please. Type it. I will read and reply to you. Guy, can we move on? Can we move on?
Yes, sir.
Okay, let's move on. I always say never forget to read pair. This mention, I circle it here. Very important, very important. Then click here. When you see the pair end, you come, you see the round. There is somewhere at the level of the foot, last part of the page. You click on this accession number. Let me go and do it with you. I am here. I come here. We click it. What do you see?
Sorry, you've stopped
We
sharing
see.
the screen.
No, I'm still sharing the screen.
We can't see your screen.
You can
The
see.
screen is shared. Maybe some internet connection issue. Screen is shared.
Perfect. You remember this place, right? We did the same with single end. You click in fast day. That is something important. When you click, this message appear. This run exceed the download limit of 5 giga base. Don't care about SRA tool. It is another way to download, but I will explain to you later when you will be good at bioinformatics. I will explain to you how this thing works. Just this information. When you click on FASTA queue download, he said the run exceed the download limit. Who can tell me why? Why this why does this message appear?
Data is too much large, greater
Yes.
than 5 GB.
And why is it so large?
It is a pair.
Both
There's
ends.
2 reads.
Yes, because when you have pair, it's like the amount double. It's double. You double completely because the forward and the reverse have each one the same number of data, have the same size. So if you upload, it will be very big. That's why it's doing that. You see that with the single end, we could download from by clicking on FastQ. Let me explain here. This message. So we cannot download because it's so big. But when it was the single end, we did not have that. It went quickly. When we click here, he brought us here when we click on download. I don't know if you see. When it was the single end, when we click here, it was done. We did not have to when you click here, it was done. But with the pair end, is saying that this, we say it's because it is very big. Move your mouse on metadata. Click on metadata. I went to the metadata. Are you there?
Yes, sir.
Yes, sir.
Okay. And in the metadata, see the arrow, guys.
Yes.
In the metadata, you will see run. Copy the accession number SRA, blah, 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 that I circle it. Run. You copy it. Are you there?
Yes, but it's the same
Yes,
number.
sir.
Yes, it's the
Yes,
same.
yes.
It's normal.
Yes.
Don't care. Just copy it. Now you know that it's the same because I told you that it's a single end. It is pair end. Someone like me, I would have never come here because I know that if it is pair end, I should use European Nucleotide Archive, another database. But I wanted that you understand that you will not be able to use SRA to download double end. And for the interview, someone can ask you, how do you download single end compared to pair end? How do you do it? You should be able to say that to explain what we are doing. So I'm taking time to explain because it is important. So when you take the accession number, you copy it. Now go in the browser, you type ENA. It's European Nucleotide Archive. It's where, but we will use the accession number that you just copy to go into another database to find what we want. Is it okay?
Yes, sir.
Guys, if you are happy, you just give a thumbs up. Let me post first because I don't know if we are together or not. If you are satisfied of what is going on,
So why we do this?
give a thumbs up.
Why we do this? Download
Pardon?
from why we download it from another website?
I don't get you well.
Why we download it from another, not CPI from the European database? Why we do this?
There is different way to download, but the first database that you should, for most of the sequencing RNA-Seq, when it comes to sequencing data analysis, always start by there is SRA, but there's also GIO. There are two databases that can be used. There's GEO and SRA are the two main databases where you will have immediately. With NCBI is like the big umbrella, and other database are just under NCBI. I don't know if I answered your question. Hello? Yes, because when you go into the NCBI, it's where you, when you go into NCBI, it's where you now choose the database that you want to move on. Yes.
Okay.
Okay. Are you ready, guys? Can we move to the next step? I say you must type ENA. You Google it. ENA browser. ENA as database. Hello? Hello?
Hello?
Who is? Guys, browse ENA browser. Are you there? Are you here?
Yes.
Yes, sir.
Okay.
Yes.
Click on it.
Yeah.
Click on it. Something will appear. It's European Nucleotide Archive. Is it okay?
Yes, sir.
Can we continue? Okay. The
Yes.
accession number that you just copy before opening ENA, come and paste it here into the accession number here. And then you click on view. Let me show you. You copy this number, right? Then you open, in one, you copy this number, in two, you open ENA. In third, you come and you paste that ENA where it is called accession number. Then you click on view. 3 step. You copy the accession number. You open ENA database.
Hello?
You see, oh, who is making this noise? Please respect the class, respect people. If you are busy doing something,
Yeah. Please,
yes.
I have a question, please. Can
Okay,
you,
go with your question.
please, can you show how, this is not to take you back, but I'm sorry, please, how you go to the metadata? I seem to have missed that point.
Okay, just look at the screen. Look at the screen. When you click in FASTA, see where are the red? Everywhere you see red. When you click on FASTA download. Are you here? Hello?
Yes, we hear you.
The person who is asking the question, I want to make sure that he understood.
Hello? Hello, I'm there.
Please, let be clear. Look, there is a lot of people in the class and we have to respect them. If you ask the question, I want you to be there to understand the question. You cannot ask the question when you are doing another thing. It will be disrespectful for other team.
No, I'm with you. I'm with you.
Okay, perfect. Are you here? Did you understand this place? FASTA, when you want to download, you click on download. Did you click here, download? Did you click on download? This.
Okay.
What happened? You will see the message, this run. You will see the message that appear. He said, this run exceed this download limit. In red, here. Is it what you are seeing or not?
What I'm seeing here is, I don't know if I'm still on the old one, because it's still, I don't know if I'm looking at just the single
okay.
Sequence.
It means that you are in a single. You can have, when it is pair end, you cannot escape this by this message. It's not possible. It means that you are
Okay.
you are behind us. What we will do, I will
Yes, that's
come
what it
later.
means.
I
Yeah.
will come later
Okay. Yeah.
and work with you because I don't want to
Yeah.
block all the class. Let's finish this part.
Yeah.
We should finish. Okay.
Okay. Thank you. Thank
Yeah,
you.
I will
Sorry,
come
sir.
later. When we finish this, I can, yes.
Sorry, before you continue, sir, kindly help me with the accession number on chat box so that I will not disturb the class.
You can even copy it here.
I can't copy.
SRR, you can see it. Okay, let me send it to you. Guys,
Thank
why
you.
are you giving a lot of job to your teacher like that?
We are very sorry, sir.
Why are you giving a lot of job to your teacher like that? Oh, okay. Let me send it in the chat. This is the last time in the chat.
Sir, I just want to say one thing.
Yeah.
The session number that you shared right now is not the exact same in the presentation. So that's why.
Yeah. That's why the accession number I have in the presentation, because I extract it, now I started looking at it, I was not seeing. That's why I went online when we were working now to get this one. You can get any accession number and try with it. This one I just picked this one online because the one I wanted to share with you, I'm not seeing it, and I don't have time to go through different file and look at it. That's why I just say better I pick any one randomly and share with you, but it will be the same. It should not be a big issue.
Yes.
So use this one. You change after that and use another one. Okay.
Okay, thank you.
It's the same rules. Accession number doesn't change the rule. This one I just pick it with you right now because the one that I wanted to share with you, I don't know where I put it. I did not put in my slide. I thought that it was in my slide, but it's not there. That's why I just picked this one online right now. Can we continue? If someone has a question, the person can ask, please. Ask all your question, guys.
So when I open it in European nucleotide archive, so the result is ENA base count 0. ENA is Y. I don't know.
Let me go to it. Okay. When you click
So
on,
when I copy the section number, I copy it from the chat.
Did you copy the same identification that I just shared? Copy it. You go to ENA. Let me do it. I will do with you. You know, let me do with you. You see what will happen. I open the ENA, right? Are you here?
Yeah.
You come, you paste where there is accession number. You have enter the text, you have accession number. You paste where there is accession number. Then you click on view.
Yes, and then you click view. Yes, it's like this.
Yes. Okay. That's why there is something. That's very good. I understand. Normally, it means that the data they did not give. Normally, with the accession number that I took, we should have this. We should have all this detail. We call that the metadata. It means that they did not load everything. It's not my problem. It's not yours. You can do anything right now. Do you understand?
Yeah, I do it another different number, so it's give me the summary of the study.
Okay. Yes. Yeah, it's the summary. But you will not be able to download your data here because see that it's not giving you all the information, but try it. It means that we will end up by using this NRA or this one. I did not want to type, but we will do it. Look, but that's good. That's good what is happening. You see that there's no way to, okay, let's see download. You see that in the downloading, that is a very good problem that we are having. When there's no way to download, it is giving you XML. This is not a good file. You want to have your file in FASTQ, fast queue. There's no way to have FASTA or FASTQ here. It means that we cannot use this one. That's very good. That,
so we
now
need in this section it's something. So we need in this section download FASTQ file, not Excel.
Never. Always. That's it is a must. All the raw data must be always in FASTQ. Why? Because I say the FASTQ has all the detail, like all the quality control information that will allow us in the quality control step to check if the sequencing was well done or not. It must always be in FASTQ, always. So here we don't have FASTQ, we cannot download this. It means that this data is incomplete. It's not because of me, it's not because of you, it's because of the person who worked in this project. They did not upload all the information. So you and I cannot use. That's why the data that I used before, let me copy it. I did not do, I was a bit lazy to copy it. I extract this data. I don't know where I put it, but let's try to, can someone copy it for me? Let me, I take a pen to copy it. That's a very good observation. It's good when we have some time problem. If everything works, we will not know what troubleshooting means. Guys, 1 minute. I copy this accession number I share with you. 14 6273. Okay, let me share with you now, guys. Let me type first to make sure it is oh, it is SRR 514. Is it that? 6273.
Yes, you are correct, sir.
Is it correct?
Yes.
Because
Yes.
if there is okay, let me share with you. It will allow us. That's very good. The problem that we just had, it's good to have this kind of problem. Let's paste this one, but the process is still the same. Okay, you see this? Okay, this one, stop here. Look, you don't have something, but if we just paste what we just copy, we click here, you will have download here with FastQC. You have all this information and you have this part, read file that exists. And the information we are looking for is this one. In the order file, we could not get it. In the order, we could not get it. We did not have a read file. We just had it was incomplete. So we could not use. But contrary this one, we have all the information. Is it clear?
Yes, sir.
Okay, you come here. So we are here. You move. Let me put full screen. You come here. This is what you are having now. All of you must have this page.
Yes.
Okay. You come here. You have download all. You'll see that you see generate FASTQ file. Dot. You have file transfer protocol. Don't care about this one. I will explain to you. Before the end of this class, we will do it. When you see fast FTP, it's, let me show you right now. First, click here. When you see FASTQ, click first, you will see that you have the accession number /1 FASTQ GZ, and you have accession number 2 FASTQ. So the two that you are seeing here are forward and reverse strand. Click on each one. Click on the first one, then you click on the second. I click on the first, I click in the second, and you will see that it is downloading. Both are downloading. The first one that we downloaded is here. Are you okay?
Yes, sir.
Guys, if you are with me, let me know. If you are getting it well, let me know. If there is a problem, share your problem. We talk about it. You are here to learn, not just to look at me. Okay, happy to know that everything's going well.
Yes, everything is going on well. It's just that the download is big, so it's taking time.
No problem. That's why we
one.
just continue doing some stuff. That's why you will see that people work with very powerful machine. And it's for that reason that I told you that I don't want to go start working with people on Galaxy, because you will not learn. I prefer that you start seeing the real problem of bioinformatics from your own computer. Files are too big. Files are too big. That's why you should have let's say at least 50 gigabyte 50 gigabyte to be able to handle the smallest.
Yeah.
Is it okay? We'll be working.
Yes, it's okay, sir.
No, we HPC is a very technical skill. Oh, it is very, very, very advanced skill. Very, but and it's very expensive. You will see the high performance computer more often in the cloud for people who can work in the cloud, or in the big company. You will not see someone like you and I having HPC for what? It's very expensive, very expensive, very expensive. But if you can see in different in the university, the bioinformatics, maybe the genomic platform of the university, you will have this kind of stuff to run big amount, large scale data analysis. Or yeah, is it okay for everyone? Can we move on?
Yes, sir.
Yes, sir, it's okay.
Okay, someone is talking about the recording. I started recording, but I don't know. But if it doesn't work here, you can go on YouTube and have the recording, because we are streaming the presentation also on YouTube. Okay, can we continue? Let's come back. So you have downloaded. So guys, now you can, after this session, you can say, I am able to download an RNA sequence either pair end or single end. Can you say that to me, that you are able to do it? If someone give you a session number, give you just the accession number of any sample and ask you to download, will you be able to download?
Yes, sir.
Yes, sir.
Yes.
Guys, okay. If you say no, we can re-explain, because the only thing that can make me feel good is when I know that you are understanding it, you are getting it. That's the only thing that can make me feel happy. If you all understand what is going on, give a thumbs up, guys.
My challenge is for the double. Yeah,
I
my
will come
challenge is
to
for
you.
for download the paired end. How to get how to get the accession number. Yeah, thank you.
Yes, let when we're finished with order, I will come to you. After this one, we will finish with order. I will take 30 minutes just for you.
Okay, thank
Is
you.
it okay?
Yes, it's
But
fine.
listen
Thank
to
you.
what we are doing. We have finished.
Yes, I'm following. I'm following.
Guys,
Mm.
now let's go back to what we call WSL because it is the fastest way and the more technical way to work. It's what you will see in the company. In the company, at the university, people will go manually like this. But in the company, you should be able to work with command line. Here we download it manually, but we should be able to do it with the command line. That's why I come back. Don't change. I come back with this concept. Very important concept. The file transfer protocol. You saw that they say here generated fast queue and they put file transfer protocol. Right, guys? Are you seeing FTP?
Yes, sir.
Here.
Yes.
Okay.
Yes.
Okay. If you are seeing FTP, keep there. Let me go to come put your mouse immediately in the on the forward brain and you make a right click. You will see something, copy link address. See exactly what I did. You put your the mouse in the on the accession number, you right click and then you type, you copy the link. Something like copy link address will appear. Are you there?
Yes, sir.
Do that for the the reverse strand as well. Is it done?
Yes, sir.
Okay. What I suggest you open a notepad like this one. You paste there. Open a notepad, you paste what you just copy. You will see something. You can see my screen. Are you seeing my screen? This part in blue.
Yes.
So okay. You will see that this is the link to access your data without going manually. That's why if you will see that this comment, it's exactly this one. Exactly the same. When you want to download in the Linux without handle it manually, you must do the same step that we did, but instead of downloading manually, you will just copy the link that will have the, the file transfer protocol. You saw how we get this one? Did you see how we get this? If you did not see, we go back again. It is very important what we are doing. Because after that, we will start doing the quality check. This. But this part is fundamental to know how to download manually or by using the file transfer protocol. It's this one. I come back again. I repeat so that everyone.
Can we go back on the FTP again, please?
I came here, I said that you are seeing fast TQ file. Column, you have fast TP, FTP, file transfer protocol. it is, and why this? If you come here, you just put your mouse in on one of the anyone, you right click, something appear like this. You come here, you copy the link the same way you copy the link of your LinkedIn. The same way you copy the link. When you copy the link, you open a notepad or a Word format. It depends what you want. But I suggest you to open the notepad. You copy it. So you paste there. Very simple. You do that for both first brain, forward and reverse. You must always do that for both. If you have pair N. Is it okay? Guys, if you say nothing, if for me, it means that you did not understand anything.
Yeah, it's okay.
Mariam, is it okay?
Yeah.
It's okay.
Okay. Charles, at least did you understand what we did?
Yes, it's clear, crystal clear.
Okay. Now, we must, because we did everything and now we are going to use the command line to do the same thing. And it's the same way it works with hash HPC. What we are going to do now, it's what people do with hash HPC. But the difference is that HPC is more powerful. It means that it will download faster. If for your personal computer, it takes like 2 hours to download, with HPC, it can go in 2 minutes. That is the main difference. But the principle, the command are the same. Is it okay? Now, let's go back again. We must install this guy. We must install this guy today and download at least one sample, so that next week we will be doing quality checks and other step. Do you want me to go further? Okay, someone has a question. Ask your question.
The WSL, after completing the download with command protocol, it says that changes will not be effective until the system is rebooted. So it's a must.
Till the system is... Oh, yeah, to reboot. Yeah, it's true. It means that you should start. Yeah. Okay, guys. Since I will do something, you will see. Since you have to reboot the system to have your WSL working. I did that myself. I did that as well. So you have to do that. But see something that I'm going to do. Are you seeing my screen?
Yes, sir.
Yes.
Okay. We will repeat that. Maybe I will share the video with you. You will watch and try to install WSL today. If you don't succeed, tomorrow I will schedule 30 minutes or 1 hour so that we will manage that. Because I want this part to be done this weekend. We should install and be able to download both manually and with WSL. It should be done even if we are late. It means that tomorrow we schedule another class to finish it. Do you agree? Do you agree, guys?
Yes. Yes.
Yes, because I want that each day, each week, we achieve our goal. And if we are late, we reschedule. Because I can start rushing, but I don't want to rush. I want you to understand, to be able to leverage what you learn here in the real life, in the real scenario at work. That's why rushing is not our goal. Is it okay?
Yes,
Yes.
sir. Thank you.
Okay, just see what I'm going to do now. I will share this command with you guys. Don't worry. We just download this one, right? This is, you can see the accession number is the same one that we pick from the, I can do it again so that you will not say that I am doing something that I come here, I copy the link, right? I go in my notes. We can even open a new one. No, I will delete. Let me paste it here. When you install your WSL, you reboot it. So you start the computer again before it start run, before WSL will start running. What we will do now, I tap wget, wget c, I paste, I go, I pick up the, you will see something. This is the link, FTP link that we have downloaded. I copy it. I come, I just paste. Do you see what I did?
Yes.
Yes.
What I did, I just ran WC. So wget means web get. So get from the web. W that you are seeing is web. Okay, guys?
Okay.
I want you to understand any single detail. I don't want you just to use without knowing what does it mean. What did I say? Wget means web get. It means that you are going to get. What? Someone wanted to ask a question.
Can you send the accession number of study? The accession number of the study.
Yeah, you. I don't get you well. Hello.
I want someone to send me the accession number of the study.
I will send you. I will. It is in the chat. Can someone send that in the? Can someone share?
I
with
will.
me?
I will.
Please.
I will.
Guys, see what will happen now. See what will happen. This is a Linux command, but I am running it from my Windows. So when we install SL, it allows us to get our sample from the web. That's why we are giving that instruction. Get my sample from web. That's why we say wget. What does C means? C means continue. It means that if it happened that, let's say the electricity issue or the internet issue happened, and the process can no longer continue because it is interrupted accidentally. When you will start your computer again, you will not start over. It will continue where he was stopped. That's why you put C. But if you do, we can also download without putting C. It means that if we have a problem, we will start the process from scratch. So we can remove C, it will still work. But if something happened, we will start from scratch. But when you put C, if a problem happened, Then when you restart again your computer, it will continue where it was interrupted. Is it clear?
Yes, sir, it's clear.
Yes, sir.
Okay.
Very clear. I really
Okay.
appreciate this information, sir.
Yeah. So C should not bother you. Some people will run without C. You just understand, the person did not care if something interrupt. He will have time to start from scratch again. But you, if you care, you don't want to lose what have been done before, you put C to just continue where it have been interrupted. Okay. When you is like that, you just press enter. I am not longer using the mouse because it is a command line. You will be working with your hand. You press enter. You can you press enter. See, you will have the same. See the percentage. Now, since I order to go in the web, get the accession number from the web. So and when you say that, you give the link. The link, it's like the link that allow you to get into a website. You share the link where in the web the folder is located. So you give the instruction to you give the instruction via WSL that go in over internet, and you fetch this information and you give the link. It's like the address. You give the address and you order it. You say, go and find it for me. I don't want to do it myself. Go and do it for me. You see, it's doing the same thing that you and I was doing manually, but we are doing it in the command line. And this is why I tell you that you must learn Linux. You don't have any option. If you want to say, I can work in the command line and work well, easily, you must know Linux, because I'm sure that a lot of you have never seen this kind of thing. Have never done. But you will do it. I am showing what we are going to do. I am not the one who will run it for you. You will run it yourself in your own computer. Or is it what you want, guys? Is it what you want or not?
Yes, that's what we want.
Other people don't want to say something. Guys, is it what you want? You want to work in Galaxy, or you want to be able to work from your personal computer? Okay. You can see that it is downloading. The same way that we download by clicking, we will have exactly the same result if we download our file. But now you have started, you can go and eat when it is running, right? But to do that, we must install the WCL. Do you want me to install the WCL now? We have finished our class. Normally, it's a 1:30, 12:30, but we can take 30 minutes, we install.
So
do you want?
For me,
yes,
I've restarted my PC, but
yes,
the WSL is not opening. I don't know why.
Okay, I think that let's schedule tomorrow to be exclusively on WSL. What do you think, guys?
It's okay, sir.
Okay.
I want
to ask you.
Yes, a few seconds,
So I want
guys.
to ask.
A few seconds.
Okay.
Let me go back. Where am I? I want to make sure that everything we are doing, all of you, where I want to go back to our, okay, we are here. Okay. Yes, someone were talking. Someone wanted to ask a question. I don't hear you, guys.
Can you hear me?
Yes, it's good.
Yes. From the ENA platform, the
So
plugin.
ask me about.
Can you type your question in the chat box, in the chat? Guys, if someone has a question, the person can type it. If someone
So,
has a question, you can type it.
sorry, I think it's better I ask because it will be long a little, sir.
Okay.
So, my own downloading has completed from the ENA, from the European platform. How do I take it to the Linux?
It's the same like, no, oh, I understand. You will put it in a folder the same way that you download something or any file in the internet and you organize in your, it can be in your desk, in your desktop or in the document you put in a folder. Now you want to get access to it. Okay, let me show you our, but it is a very, now you want me to go into Linux to work with file. Normally it is a skill that is purely Linux skills. Okay, look, in Linux when you type like that ls, it means list. Here for instance, okay, that is something that I downloaded last day. All you see, when I type like this, it gives me all the file that I have. If I have downloaded something by typing ls dash a, it's like list all. You see, these are the file that I have. You have found the file here. You see, and if you want to open a file, I have not yet downloaded anything except this one. So if you want to open it, you type cd and you put the name of the file. So ls, to get the name of the file, you type cd and you copy the file and you paste here.
Okay, it's clear, sir. Thank you.
Yes. Tomorrow we will do it anyway. When we will download, we will organize it and manipulate everything without going as without clicking as we often do with the mouse. We will be using our finger to get access. That is what we call file manipulation, but it is another skills. That's why I say that you must learn Linux. It is not an option unless you don't want to be bioinformatician. Linux is a key skill to learn because what we are doing now is Linux. What we run, what is going on now is Linux. You should know what we get means. Wget means. You should know why we put C. There is another way to download. Have you heard about curl? Curl. It's C-U. Let me type it for you. Curl. Have you heard about curl? Curl. If you type curl versus
No,
curl
I've
means,
not.
yes,
Okay.
it is another way. It's client for URL. It is another way to download. It works the same as
We get.
WebGet.
Okay.
WebGet. But for Linux, you will see a lot of people using this one. I will not go through it. You can read advantage and limitation. You can do it yourself. But we could use curl. If someone wants, the person can try curl. It will work as well. The same thing we have done. And you can see here, does not build in recursive download functionality. I will explain to you. I understand this very well, how it works. But right now, just think about WebGate. Okay? Don't look at curl. Forget about it. I did not say anything to you. Is it okay, guys?
Yes.
Hello? Okay. What since today you are 30 something, we have to, okay, let me answer. Should the load data to in the terminal? Yes, you can run both data. I suggest you to run one. When you finish, you run the second one, because if you run both, it will use your CPU and the memory. Your machine will slow down. Run one task. When you finish, you run the second. That's why for this training, we will not be downloading the whole genome. We will download the chromosome to work with, to avoid having the whole genome that will be very heavy to manipulate. But it's the same we do in the lab. In the lab, we work with the whole genome. But since we don't want to work with a lot of data, the performance of our computer are not very powerful, we will just take a chromosome to work with it. Okay, guys, I think we have done the main thing of the day. I will share a link for tomorrow again in my on LinkedIn, BioDigitera, and also in the Facebook account. I will share let me share the Facebook with you guys, and I would like you to like. Let me share the Facebook account of BioDigitera. You like it, right? The only thing I can have from you.
Yes, sir, of course.
Let me first. I am a bit confused. Okay. Just you like it and you share. That's how you can help me grow as well. I know that a lot of you doesn't know that this page exists. Okay. I share it with you. You just like it. And also you can go in the on LinkedIn, not on LinkedIn. I share with you guys. Like it for me. Then I have also my okay, I think no one has question. Right? There's no longer question, right? If there's no longer question, go and like, and we close the session. I will share the new link. Tomorrow we will be work. There is this guy who wanted to work in the comment line. There is one person, I told him that I will explain to him. Guys, if you all like the page, we can move on. We can stop. Okay, Charles, we will stay together. I will help
Thank you. Thank you so much. What
Charles,
time are we meeting tomorrow, sir?
I will share, I will publish something online, maybe at 10 or maybe, yes, 10, because we should take in account Indian people at 10, yeah. Guy, like the page, and you like also the YouTube, please. I will share everything online on my LinkedIn page and in the BioDigitera Digitera LinkedIn page. And if we have time tomorrow, maybe we will do again 2 hours, install and go till QC. maybe. So tomorrow, think about 2 hours, maybe. Do you like the session, guys?
Yes, very interesting and very educative.
Very much. Yes, sir. Thank you so much.
If you think that the transmission is not good, criticize me. I improve with you.
You are doing well, sir.
Okay. If you think that the transmission mode can be improved, don't hesitate to tell me, because I'm here to grow with you. No one is God. We all learn from other people, okay? And believe in you. If you want to become bioinformatician, it is a very booming field now. Bioinformatician is a booming field because we are having a lot of biological data, and people who are in Africa and in India, you have a lot of data to work on, a lot of topics. So give energy to acquire the knowledge. Okay, someone is asking about recording. I will see if I will see. But I said you can watch it on YouTube as well because it was a streaming session. And I hope that in YouTube it will stay there as well. Okay, guys. If someone has a question, the person asks. If you don't have, just let me with Charles. I have to take time for Charles. Charles? Charles, are you with me?
Yes, we are here.
Okay. Okay, people who did not understand can stay, and I will go back again. Okay. Charles, what was your question? And Sarah, what was your question? I want to understand. Since we are two, I think it will be fine.
Yes, I'm here.
Yes, tell me, what was your question?
Yes.
What was your
I
question?
said, yeah, it was on how to get the accession number for the pair end sequences.
Okay, no problem. Let me give it to you.
Hello?
Let me give it to you. It is somewhere.
Okay, thank you.
I paste in the chat right now. Let me check to make sure that it's the right accession number. 73, yes. I just sent it right now to you. Take it, you go in, we will start from scratch. Let me make sure that people who are still there has the same problem or not. Sarah, are you there? Okay. Anyway, let's go back. Let's go back. For the pair n, first, you go in NCBI.
Okay.
You choose, you will see all database. Do you see all database? Here, you will see somewhere, you click and you go, you select SRA. Just look at what I just circled. When you choose this, you come, you paste now the accession number that I just sent to you in this search bar.
Okay.
And you click search. You click on search. Are you there? Let me
Yes,
stop.
I'm there.
Let me stop this session. I am still
My
working.
it's loading on my laptop.
Okay. And as I said, when you click here, you always check to make sure if you have a single pair, the pair end or single end, you check what are you seeing. It should be pair end, right? Here, you check. This page must appear when you click on it, when you search. Is it what you are having? Can you share your screen with me, please? Can you share your screen? I don't know if you are online or not. If you don't talk, I feel like I'm alone.
Okay, yeah. Mine has opened. I'm seeing the search results 1 to 20.
Yes. No. It's not what I told you to do. For sure, you did not do what I said. I am 100% sure. You did not say, you did not do what I said. You are not doing. I come back here. I have everything in front of you. You can just look at it. Are you in? I said you paste the accession number in this search bar. Did you do it?
Yes, I went. Yes.
Share your screen, please. I stop mine. You share yours, okay?
Yes, that's what I did.
No,
Can I
please,
recount what I
please,
did?
please,
Yeah.
please share your screen.
Let me go back. Let me.
I
can't. I'm doing it on my laptop, and I'm using my phone for the.
You mean that I can help you? Because if you cannot share your screen, I don't know how I will see where you are doing mistakes. Maybe you are doing mistakes somewhere. I have to be able to see your screen to help. If I can see your screen, I will not know how to troubleshoot and understand where is the mistake. That's the big problem. So since there are some people online, let give the, let maybe some people has, let me share.
Yes.
Look, if you,
I'm unable to share my laptop screen because it has.
Yeah, I understand, but I cannot help you if I cannot see what you are doing, because I have everything here in the slide, very well explained. If you don't understand what is, I will share also the slide with you guys. But if someone is having a problem, the person can share the screen with me. I go through the process with him, explain, see what the person is doing, and help. If you don't share your screen,
Sir, can I share my screen so that I can use it to learn?
Okay.
So you stop sharing so that I can share.
Yes, I understand.
But first, I want to know. I don't want to that you monopolize other people. There are other people who are still online. Do you have some concern you would like me to help? If yes, let me know. I will know how long I can give to. I can stay with Charles and give the floor to another person, because I want to give the same amount of time to each of you. But I have to make sure that all of you are here for the same problem, or you have another question to ask. Okay, I will consider that Charles is alone. Charles, I give the floor to someone, the person will do and help you. Okay, share your screen, guys. Share your screen.
Yes, I'm with you. Okay.
I have not seen your screen. Okay, perfect. Okay, click on search. Okay. After clicking on search, there is something that I will say best practice. You must always check to know if it is pair end or single end. What are you saying? Pair
I'm
end.
seeing paired end. Yeah.
Okay, pair end. Go
Yes.
scroll down. Scroll down. You see run. Click on run.
Run.
Yes. No, click on you. You are seeing the accession number. After run,
Yes.
yes, click on it.
Yeah.
Okay. Then what happened? You will have faster queue, a faster/faster queue somewhere, just on top.
Okay. On top.
No, on top.
Yes.
Yes.
Yes. Faster.
Click.
Faster queue download.
Yes, click on it.
Okay. I don't want to download again.
You will not download. You will not download.
Okay.
There is a message that will appear. There is something that will appear.
Yes, that's
No, here you are working with single end.
is it single end?
Okay, click on fasty. Click on fasty now. Down. No, no, no. Down. No, fasty queue. Fasty queue. Fasty queue. Something will happen. Is it able to download? It is downloading.
It's
It means
downloading.
that
Yes, so it's downloading.
no, I gave you the wrong thing. No, we are not dealing with the same accession number. Let me see. Because you should not be able to download the. Did I give the same accession number 73?
Yes, it's the same accession number, sir. It's the same accession number.
Let me go back. Let me go back. What did I do? Let me make sure. Okay, let's see.
I should go back.
How to do and click this accession number. I will troubleshoot. I am seeing something that is bizarre. Click this. Let me make sure again because I am a bit 514. Yes. Click copy this link. I just want to show him, then copy this link, go into ENA. We will come back. I will come back. Let me try something myself. I am seeing something before talking. I have to go in ENA, show him exactly as you did. I want to do something right now. NCBI myself.
This is ENA, sir.
Okay, ENA paste the accession number there.
Okay.
You will paste where on top. There is two search bar. There is accession number. You paste there and you click view.
Yes, that's what I'm doing.
What are you seeing now?
I'm coming.
When you click on view, what will appear?
Yes, you can see what is showing. It shows
Okay, you are having all the
information. Yes,
all the information. Okay, good.
Yes.
Go down, scroll down.
Yes, I'll scroll down. I can see download.
Yes.
Okay.
Normally you should have, what is going on? You should have the two data appearing.
Yes.
Click on download and we see. There is something wrong going on. There is something wrong going on. There is something wrong going on. Let me do
Yes, I
it.
remember the first time we did it, we saw the two datas.
Yes, there is something wrong going on. Let me do it. You know, but it's not us the problem, you know, because you don't, we did it. Let me type the, let me go back to my, the session number is, let me make sure that it's the same problem. If it is not a region related, because sometimes it can be a region related issue, I don't know. Let me do it.
I want to, I agree with you, sir. I think it, but we did it too this afternoon. Now
Yes,
they said
sometimes,
it's unavailable.
yes, sometimes it might be a region related issue, but let me check to avoid saying something just, okay, I click here. And I'm having exactly the result. Okay. I am seeing something very mysterious, but it's fine. I will explain. Okay. I will explain something to you. That's good. There's something that I'm seeing, but I will come to you. There is a detail that I forget to explain, but you just, I am in INA. I click on it. I copy the accession number. I paste. But let me show you how it's appearing here. There is a problem. Guy, let me take the control to explain. I am having exactly what we were having. Let me take the control and explain. I think there is something going with, according to the location where you are, we can have some issue. Can you give me can you let me share my screen?
Yes, I have stopped sharing, so you can share, sir.
Okay. Here, I just did it again. Are you seeing my screen?
Yes.
Yeah, I can just delete. I start ENA in front of you so that when I am talking, it's not you. You will see that sometimes it can be region related issue because we are having, I paste here, right? I click on view. What happened? I'm seeing here, but it's not what you are having.
Yes, maybe it's region related.
Yeah.
But I also have this initially, and I downloaded it too before.
Okay. Now you did something that I forgot to mention. You will see that when you click here in NCBI, you click here without going in ENA. It started downloading.
Yes.
Here. But what you will see that it will not have the difference when it download. If you don't go into ENA, it will just give you one brand, one strand of RNA of DNA. It will not give you two. It will not give you two, because when you download it, you are seeing just one is downloading. It's just the forward strand. And it is a detail that I did not mention. So you must always go to ENA, because with pair end, you are able to download, but it gives you just one strand. That's what you are seeing. Only one strand. That's why you must always go to ENA to have both strands here, and you click on each one to download it. or you take his link by using the protocol transfer system as we did here. Is it okay? Is it okay?
Yes, it's okay. It's okay.
Okay. I don't know if Charles now understand what we did or not.
Yeah, I'm following. I've gotten to where you are now. I'm following. It's clear.
It's clear?
Yes, it
Okay.
is. It's clear. Yes.
Okay.
You
Now,
know, when I did
maybe
the search,
other people
yeah,
has
okay.
gone. Sorry, I interrupted you.
Yeah, the mistake I was making was when I did the search on the NCBI on the SRA database, I was putting a space in between the SRR and the numbers 5. So
okay.
So when I now join everything together, it now worked. So it's fine now. Thank you.
Perfect. I don't know if someone has another question. Does someone have question? Okay. If no, we stop sharing. Pedro, you have any question?
Yes. No, I'm good. I'm good.
We have another one. Rehab. I don't know if I am calling well the name. Okay. It means that all of you are good. I want to stop now the work. What is it okay, guys?
Yes.
I want to stop sharing. See
thank
you
you.
and
Thank you, sir.
see you and tomorrow. Goodbye to everyone.
Yeah. Bye. Thank you.
Thank you.